presentation of the day. Pleased to introduce Stephane Boissel from TxCell. And we keep hearing about turning on the immune system for oncology purposes, but apparently it can go in two directions. So tell us about that.
Sure. Well, thank you very much, and good afternoon to all. I'm sorry to be the last before, wine and beer, so I'll be very quick. And I will start with a few slides, and then we'll take some questions from you, Josh. So TxCell is a French cellular immunotherapy company. And what makes us different from most of our peers is that we believe today to be the only company in the world to be exclusively focused on what we call antigen-specific regulatory T cells. And we are using those cells not to fight cancer or infectious disease, for example, but very logically, given their mechanism of action and their function, to fight autoimmunity, inflammatory disorder, and transplantation rejection. We are a platform company, and potentially this technology can be applied to fight a vast panel of different diseases. And actually, we need three things, or mostly three things, to start a program. We need to identify in this disease an unmet medical need, and as you all know, there are many unmet medical needs in this disease. We need then to make sure that there is a relevance to use Tregs in this disease. And last but not least, we need to identify a right antigen to address the disease. And very importantly here, the antigen does not need to be associated with the disease itself. It has to be locally present in the inflamed tissue. We have two platforms of product. The company was started 15 years ago on the left-hand side platform, which is called Astria, which is more or less on hold today. In this platform, we were not doing any genetic modification to the cells. We were using what we call naturally antigen-specific Treg. It worked well. We went up to clinical validation in Crohn, but we realized at that time, that was 2 years ago when I joined the company as CEO, that the manufacturing process was not exactly scalable. So we've decided to progressively shift the effort and the resources of the company on the platform, which is on the right-hand side of the chart, which is called Antria. In this platform, we are still using Treg, and again, we are only a Treg company. We are still going to look for antigen specificity, but instead of looking for natural antigen specificity, we are going to introduce the antigen specificity via a CAR. So basically, it's a CAR-Treg platform. And as you can see from the chart, the CAR construct is not very different from what you can see with CAR-T that are regularly used in oncology. Today, the technology is autologous, and we have a manufacturing process which is very similar to what you can see or expect from a CAR-T in cancer. And we no longer do any manufacturing in-house. We have two CMOs, one for the production of lentiviral vector, and the other one for the manufacturing of the cellular therapy product per se. Just a few words before we turn to question. Our most advanced program is in transplantation, solid organ transplantation, and we expect for this program to start a first-in-man study by the end of 2018. If we reach that objective, we'll be the first ever company to start a clinical trial with a CAR-Treg program. So we are very excited by this program, which is based on data that were published a year ago by a group at the University of British Columbia in Vancouver. That was the first ever proof of concept in mice of a targeted HLA to CAR-Treg in a transplantation setting. And as you can see in this slide, if you look at the chart, the survival chart, you see a clear difference between mice for which a GVHD setting was created that received no treatment. This is a black line that survives something like 20 days at max. Mice receiving a polyclonal, so non-specific Treg. This is a blue line, the control Treg, that survived longer than mice receiving no treatment, but all died within 30 days. And if you take a look at the red line, which is the line at the top, that is a line, the survival line, of mice receiving the CAR-Treg specific to the antigen of the graft. And as you can see, there is a very, very clear difference, not only between the mice receiving nothing and the mice receiving the CAR-Treg, but also in between the mice receiving the polyclonal non-specific Treg and the mice receiving the very specific CAR-Treg.
Well, thank you for the overview. Maybe to start, I think we're pretty comfortable figuring out for oncology purposes which antigen one targets. How do you sort it out for immunosuppressive therapy?
Yeah, well, the way, maybe I should start with the function and the mechanism of action of these cells, because that will, answer to your question. Well, the cells, the regulatory T cells, they have two main functions. The first one is to maintain tolerance to self-antigen, and the other one is to control the induction on the proliferation of pro-inflammatory T cells. And the way they work, you have to bear in mind that these cells were discovered like 20 years ago, so fairly recently. The way they are supposed to work, and at least two of their mechanisms of action today is elucidated, is that they deliver anti-inflammatory cytokines upon activation, and they also act through a direct cytolytic action against the pro-inflammatory cells. So to come back to your question, what is important for us, given the fact that those cells have the properties to home where there is inflammation upon injection, is to make sure that they are activated in the presence of the antigen. And again, the antigen has not to be necessarily associated with the disease. What is important for us is that it is located in the tissue where the inflammation is and where we need the action to be triggered. Okay, and in that sense, it's a limitation that we don't have compared to the CAR-T, for example, where you have like 20 identified, addressable antigen today. Here you can think about many different antigens. The only thing we have to make sure is that it's selectively present in the inflamed tissue and not everywhere else in the body.
And how does one identify and purify Treg cells?
Well, we, having been burned in the past with manufacturing, we have a very good idea on how to do it now. It's not very different from a CAR-T. I mean, we isolate the Treg with FACS, with beads, with equipment such as a Milteney sorter, for example, and that is routinely done with certain subsets which have cell surface marker. And one of the issues we had with the first platform of product is that the cell population we were using had no cell surface marker. So we had to go through a cloning step, which was not only very lengthy, very costly, but also not very scalable. But now with the FOXP3 population that we are using, they have CD25 among other markers, and we can easily sort them out in a few hours after they have been collected from the blood.
And how do you think about the difference between allogeneic or autologous approaches for this approach?
Well, that's a key question. My vision is that in 10, 15 years down the road, these will be allogenic, okay? Not necessarily because we need it, because we are not dealing with cancer, we are not dealing with disease, for which patients are going to die within 6 months, except maybe for transplantation. That's a different topic. But mostly because when I talk to pharma and to a lesser extent to investors, they all believe or they all want to see this technology being transferred to an allogenic technology, okay? But I really believe that for the next 10 years, I can develop a few products, I can market a few products with this technology that will be autologous products. So to come back to your question, it's not my priority today, but definitely for the next generation of product, we will be working on allogenic technologies.
And what are the challenges of allogeneic? I guess you wouldn't expect graft versus host as a risk to address. Why isn't it an earlier priority?
Well, simply because we just with this new CAR-Treg platform need to prove that it works, and we need to prove that it works in vivo in mice, and we start to prove it. And we need that to prove that it works in human. And I'm very reluctant to do something, funny with the cells before I can prove it in human.
Maybe you can talk about the disease selection process that you went through to wind up on transplant first, relative to the various autoimmune diseases that you can choose from.
Sure. So today we have 5 or 6 different programs, and the reason why we went with solid organ transplantation as a first indication is very easy to answer. The first one is that we had this proof of concept from the University of British Columbia, a group with whom we are collaborating, that were really the first clear in vivo demonstration that a CAR-Treg product can really make a significant difference. Second, we have identified lung transplantation as an indication for which, given the very high rate of chronic rejection and given the existence of with fairly documented biomarkers which are predictive of chronic rejection, we can very rapidly obtain a first proof of concept, much more so than, for example, in lupus, on which we are working, or with MS, another indication on which we are working today. So clearly, it's a combination of we have the proof of concept, and we can very rapidly go into a human trial and obtain within, let's say, 12 to 15 months of starting the human trial, having a human proof of concept.
And what are the timelines for that?
So we expect to be transferring the technology to a CMO as soon as Q3 this year, starting to discuss with the agencies, especially in the US, in Q3 as well, to make sure we fully understand what they are going to require for the IND. Then we expect to be filing the IND mid-2018, have the IND third quarter of 2018, start a first in man at the end of 2018. And again, I insist, nobody has ever done it, so that will be a major milestone for us, but also for the field. And then the phase 1 will be 15 to 20 patients. That will be open label. We will see the result 2 to 3 months after having treated each patient, so we expect to have the full picture of the proof of concept by 2020.
And when you do have that data, what is it that you're looking for to get a very clean read that the therapy is adding benefit?
Yeah, that's okay. The two main endpoints we are going to look at are the following. Did we manage to control the proliferation of pro-inflammatory T cells? Okay. And the second, which will be for us the holy grail, so to speak, will be, did we manage to induce tolerance? Okay, because on one hand, we'll have a therapy which will be hopefully replacing immunosuppressive drugs, but which are likely to be lifelong therapy. On the other hand, if we manage to restore the system as it was before, so to induce tolerance, then that will be a very, very good business opportunity.
As we think about the manufacturing scale-up, is this basically a page out of the CAR-T playbook? I mean, it seems like aside from flipping a switch on the cell sorter, it's generally a similar process.
Yeah, it's fairly similar. The first generation of process that we are going to use for the first in man is going to be probably longer than what Kite or Novartis are currently experiencing. But definitely for the next generation, we expect to be in the region of 25 to 30 days, and to have cost which will be very reasonable compared to the number I've seen in the press. And in any case, we have very little choice because we are dealing with, again, unless, not for transplantation, but for the other disease we are going to address, I'm not sure we are addressing disease that can, at the end of the day, justify the kind of price that, Kite or Novartis will achieve for their product, given the kind of deadly disease they are addressing.
Any questions? And if not, maybe just to elaborate on that last point, because the value proposition is a little different from the cancer therapeutics, what kind of pricing model do you envision?
No, that's too early for me to answer.
Yeah, please.
Perhaps you can tell us about some of your upcoming clinical milestones for data.
Yeah, well, the next clinical milestone is 2020, okay, and this is the soonest we'll have clinical data. But during the course of 2017, and again, this is totally new technology and this is totally unique. We are expecting to generate, let's say, 5 to 6 different data set on mice model, such as the one you have on the chart here. And given where we are starting from in terms of market cap, in terms of validation of the platform, I really believe, or I really expect that a compelling mice data set in any of the model we are currently working on should be significant inflection point for the stock price of TxCell. At least this is what I'm telling to my board, so I hope this is what will happen.
Great. Well, thank you so much for sharing the story, and thanks everyone for attending a really terrific day of company presentations and panels. Thanks.
Thank you, John.